Hey, and welcome to the second of our protein synthesis videos. In this video, we're going to look at the second step of protein synthesis, which is translation. If you haven't seen the transcription video, or you don't have any understanding of transcription, you definitely probably want to get a handle on that first before you look at this video. It's not going to make a lot of sense otherwise. In this video, we're going to look at how the information that's been transcribed into messenger RNA, mRNA, is then translated and used to produce proteins. And then to wrap up, we'll look at how prokaryotes and eukaryotes use different strategies to control protein synthesis and gene expression. Let's take a moment and just make sure that we understand where we are in this process. We have our DNA molecule up at the top, and that has been transcribed into an mRNA molecule during transcription. What's going to happen now is that we're going to go from this mRNA molecule to a protein through the process of translation. Whereas transcription was happening in the nucleus of eukaryotic cells, or in the nucleoid region of the cytoplasm in prokaryotic cells, translation is always going to happen at the ribosome, because that's what ribosomes do. They make proteins. While we're here, we should also understand how the genetic material in the mRNA is going to be interpreted. It's going to be interpreted in successive 3 base units called codons. So we've got the different codons in this image shown in different colors. We've got codon 1 of GUG, and then codon 2 of CAU, etc., 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 all the way down this molecule. Each codon is going to give rise to a particular amino acid. So codon 1, GUG, is going to call for the amino acid valine in the protein. Codon 2, CAU, is going to call for the amino acid histidine. Codon 3 is going to call for leucine, etc., 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 all the way down this molecule. Looking at the diagram, I can see that the brackets are a little bit off-center, and so they do start to shift down, but that is a fault of the diagram. It is not meant to convey anything. Every 3 bases is a particular codon, and each codon is leading to the incorporation of a particular amino acid in a successive chain in the overall structure of the protein. There are no spaces between codons, and each codon is 3 RNA-based pairs in length. This is important for understanding the overall structure of the genetic code, and we can see the genetic code in this table. We've got the first letter of the codon over on the left, the second one in the top, and then the third letter over on the right. So for instance, U gives us phenylalanine, whereas CAU gives us histidine, etc., etc., etc. A couple of notes of interest about the genetic code. The first is that the genetic code is universal in all biological systems on the planet. In every biological system on the planet, a CCC codon will give you a proline. These are all the same in every cell on the planet. Really, really cool and really useful as well if we want to do things like understand evolutionary history or use the genetic material to genetically engineer organisms. We're all speaking the same language. A couple of other notes about the code. The code is redundant in many cases. There are only 20 amino acids that we're going to put into proteins, and so we have 64 possible combinations in our codons. So many amino acids are coded for by more than one codon. Not all of them, but many of them. Additionally, it is unambiguous. What that means is that each codon codes for a particular amino acid. You're never going to have CGU code for anything other than arginine, for instance. And finally, we can see that it is punctuated. There are 3 stop codons shown here in red, UAA, UAG, and UGA. And there is a start codon, which is methionine, which is shown in green and also happens to code for the amino acid methionine as well. So this is the universal genetic code, and it is what is going to be used by the ribosome in order to synthesize a protein from particular amino acids. If we look at the process of translation altogether, we can see a couple of new molecules have entered the fray that we should pay attention to. Our mRNA that we produced in transcription is down here, shown diagrammatically as a sequence of RNA nucleotides. We can also see 2 other things that are coming into play. We've got our ribosome, which has a large subunit and a small subunit. And the ribosome is itself made out of a complex of RNA and protein. To separate it from the mRNA, we would call the RNA molecules that make ribosomes ribosomal RNA, or rRNA. And then additionally, we have another group of RNA molecules called tRNAs. The T stands for transfer. These tRNA molecules are going to bring particular amino acids to the ribosome in the order that they're called for according to the mRNA transcript. All three of these types of RNA, mRNA, tRNA, and rRNA, are all going to function together to produce a protein, though they all play different roles. If we look at a transfer RNA molecule a little bit less diagrammatically, we can see a couple of regions of particular interest for us. The first is where the amino acid is attached, and we can see that here in yellow. This is the amino acid attachment site. And then we can also see in gray here a region which we would call the anticodon. The anticodon has a sequence that is opposite and complementary to the sequence of a particular codon. So there are 64 possible codons. There are 61 different tRNA molecules. The 3 stop codons actually do not call for a tRNA molecule, so we're going to subtract them from our total. But other than that, inside of our cell, we've got many, many copies of these 64 different tRNA molecules, each one with a particular amino acid on it that will bring that amino acid to the ribosome when it is called for by a particular mRNA codon. If you remember back to our discussion of transcription, we break transcription into 3 steps: initiation, elongation, and termination. We do the same thing with translation, but the steps are different because it is a different process. Starting with initiation, we can see that what's going to happen is that the ribosome is going to assemble along the mRNA, starting at the front of the mRNA, the so-called 5' end, and the ribosome is going to arrange itself in such a way that the first AUG that it encounters will bring the first amino acid in. If we go back to our genetic code chart, AUG is the start codon. It also brings the amino acid methionine. So every protein that's ever made always begins with a methionine. AUG may also come up again later on in the transcript, and we'll just bring in another methionine at that point. But the first AUG is where we're going to start transcription, and we're always going to bring that methionine. Once it's arranged in that way, the next codon, in this case looking for the amino acid asparagine, is going to be available, and then a tRNA molecule with a complementary anticodon is going to bring that asparagine amino acid into the ribosome. The ribosome is then going to catalyze the formation of a peptide bond between these 2 amino acids and transfer the methionine to the tRNA that's holding the asparagine. That methionine tRNA molecule will now be empty. It will leave the ribosome. The ribosome will then move down another 3 bases so that we have the next amino acid in the chain, in this case leucine, available for that tRNA to bring in. And this will continue amino acid by amino acid in the elongation step. We can see that after leucine, we're calling for serine. Serine has been brought in, and then what we're seeing in this diagram is the production of a peptide bond between the serine and the next amino acid in the chain, the glycine. Notice that the growing chain of amino acids is continually transferred to the incoming tRNA molecule, and the outgoing tRNA molecule then leaves. We then shift again, and we continue this process until such time as we reach a stop codon. When we reach a stop codon, no tRNA molecule will come in, and so the transfer will not occur. Instead, the chain of amino acids will be released into the cytoplasm and will have produced a particular protein, or particular chain of amino acids. It's important to understand that in this diagram, we are producing an amino acid chain that's much, much shorter than a real-life amino acid chain, which could be hundreds or thousands of amino acids in length. But the overall steps of initiation, elongation, and termination are shown in this diagram, and so it's important that we understand what's happening in each step of the process. And so it's these 2 processes, transcription followed by translation, that produce all of the proteins in the cells in our body, what we would call gene expression. It's taking that information that's stored in DNA and expressing it in the structures of our proteins and their ensuing functions. As we wrap up this video, we'll look at some strategies that prokaryotes and eukaryotes use to control gene expression. We'll start with eukaryotic control of protein synthesis. If you watched the transcription video, you remember this diagram, but you'll see that we've added more elements onto it. What we've added to it are control elements. So for instance, we can see that our promoter actually has more than one region. We also have regions elsewhere in the genome that are called enhancers or silencers. And these are all going to work together to control the overall expression of this gene. We're going to attach other proteins to these regions of the genome around this gene, and the presence or absence of those proteins is going to make this gene more or less likely to be expressed. The general name for these proteins is transcription factors. And you can see them in this diagram here. In order for transcription to occur, it's not just that RNA polymerase attaches to the promoter and begins transcribing the gene, which in this diagram is actually running off the page to the bottom left here, but you need to have a collection of different transcription factor proteins present at that promoter region in order to allow for RNA polymerase to actually transcribe the gene. If those transcription factors are not present, RNA polymerase will not attach to the promoter and you won't get transcription. You can also have additional transcription factors that aren't required for transcription to occur, but make transcription more or less likely to occur through being present at this region. And so eukaryotes use this collection of transcription factors in order to control whether or not they're going to transcribe particular regions of the genome and express those proteins as a result. Prokaryotes do something slightly differently. Prokaryotes will often group a variety of genes that accomplish a particular metabolic task together behind one particular promoter. They can also have transcription factors that control whether or not a particular gene will be transcribed or not, but they'll also use a region of their promoter called the operator, and they'll produce a specific protein called a repressor protein that either sits on the operator and blocks transcription of the gene, or is not on the operator and allows transcription of the genes to occur. This is a way to control whether or not the cell is making these particular proteins that it needs for a particular task. If it doesn't need to carry out the task, then it will have a repressor protein sitting on the operator, preventing RNA polymerase from being able to transcribe the genes. But if it does need to accomplish that task, that repressor protein is going to go away, and then RNA polymerase will be able to attach itself to the promoter and transcribe the genes necessary. All of these genes are behind a common promoter. And so this structure, which we only see in prokaryotic cells, is referred to as an operon. And it's characteristic of how prokaryotes control gene expression. We don't see operons in eukaryotes because they're controlling gene expression through the presence or absence of particular transcription factors. As we move into Check Your Learning for this video, make sure that you can do the following things. Make sure that you can explain how the process of translation results in the synthesis of proteins. You want to make sure that you can explain what's happening in the initiation step, the elongation step, and the termination step, and be clear on the roles of messenger RNA, transfer RNA, amino acids, ribosomal RNA, all of those things functioning together. Make sure that you can explain how prokaryotes and eukaryotes control the synthesis of proteins. And finally, now that we're done, make sure that you can use your knowledge of transcription, translation, and the genetic code in order to determine the sequence of amino acids in a protein. If you can do those things, you're doing great. If not, that's okay too. Take a moment, pause the video, and write down any questions that you have so that you can get the answers that you need. Thanks again for watching. I really appreciate it. Have a great day.